Hello, I'm Sita Priya Murthy, the first author of the publication, The Genomic Landscape of Lung Cancer in Never Smokers from the Women's Health Initiative, published in JCI Insight. I'm very excited to share with all of you in this video the work presented in this paper. Before we begin, I would like to thank all the co-authors of this publication, the Women's Health Initiative, our funders, and the patients without whom this paper would not have been possible. So let's dive in. I would like to start off by giving a brief background, discussing about lung cancer in never smokers, and highlighting the motivation behind this study. Lung cancer is the leading cause of cancer-related mortality. 18% of deaths due to cancer occurs in individuals with lung cancer. And this very fact highlights the importance of lung cancer research. While the majority of lung cancer cases can be attributed to a history of smoking, up to 20% of lung cancer cases occurs in individuals with no history of smoking. In fact, if considered as a separate disease, lung cancer in never smokers is the seventh major cause for cancer-associated deaths. Thus, lung cancer in never smokers is in fact not as rare an occurrence one might think. Anyone with lungs can get lung cancer. Lung cancer in never smokers have some unique patterns of prevalence. It occurs more frequently in females than in males and is more prevalent in women from South and East Asia. When looking at age at diagnosis, never smoker with lung cancer is diagnosed at a much older age. And finally, lung cancer in never smokers tends to be of the adenocarcinoma histology compared to squamous cell carcinoma, which is seen more often in patients with a history of smoking. Taken together, these data suggest that perhaps biological sex and/or geographical location may play a role in lung cancer in never smokers. Thus, keeping in mind these unique characteristics of lung cancer in never smokers and others that I will delve into as we discuss our paper, the three key questions before us were: what is the genomic landscape of lung cancer, particularly in older women? Are there any unique genomic characteristics that distinguish this cohort? And finally, can we identify any endogenous or exogenous drivers of lung cancer in older never smoker women? So to address these key questions, we designed this study that is the basis of this paper. We had the wonderful opportunity of collaborating with the Women's Health Initiative to secure samples from. The WHI recruited over 160,000 women in 3 overlapping clinical trials and an observational study investigating the health of postmenopausal women. From this cohort, we were able to identify 73 individuals who developed lung cancer and consented for the study. We were able to procure sufficient lung tumor DNA and matched normal DNA from 36 never smokers, 20 light smokers, and 17 heavy smokers. This table shows the clinical features of participants included in this study. From these DNA samples, we then performed a custom whole exome sequencing on all samples. Our whole exome sequencing panel included additional baits targeting regions with known chromosomal translocations in cancer. A subset of these samples underwent additional whole genome sequencing and/or targeted sequencing to address more specific questions, which we will discuss in the upcoming slides. The first analysis of the sequencing data that I'm going to discuss focuses on the differences in somatic mutation from smokers versus never smokers. I will be showing how our analysis recapitulates some of the known somatic differences, but what I will be highlighting are some of the novel findings that we made in this study. We first look at the tumor mutational burden for each smoking cohort. Similar to what is known in the literature, we see heavy smokers have a significantly higher tumor mutational burden compared to never smokers and light smokers. This is because smoking increases the number of genome-wide non-silent mutations. This is seen more clearly when we look at the relationship between the number of pack years smoked and the non-silent TMB. One pack year is equivalent to smoking one pack of cigarettes a day for 1 year. And in this graph, you can see that as the number of pack years increases, so does the non-silent TMB. We also looked at the influence of sex and age in 3 external cohorts and found that tumor mutational burden was not affected by these. Another characteristic of smoking-driven lung cancer is the percentage of C2A transversions. C2A transversions are caused by the association of cigarette carcinogens with the DNA. And here you can see very clearly that the percentage of C2A transversions is significantly higher in heavy smokers compared to both never smokers and light smokers. And like the non-silent tumor mutational burden, we can see the significant correlation between the history of smoking as depicted by pack years smoked and the percentage of C2A transversions. In line with these observations, wherein the never and light smokers are very similar in mutational type and burden, we henceforth group these two cohorts in our subsequent analyses. The RTK RAS-RAF pathway is known to be frequently mutated in lung cancer. As you can see, however, the genes mutated differ between never and light smokers and heavy smokers. Never and light smokers have significantly higher mutant EGFR compared to heavy smokers, while heavy smokers frequently have KRAS mutations. We made a very interesting observation with respect to mutations in the tumor suppressor MGA. We found that MGA mutations were exclusively found in patients with a history of heavy smoking. Looking at 3 external data sets, we found that both in females and in males, this observation also held true and that MGA was significantly enriched in heavy smokers. We also found that older individuals had significantly more MGA mutations than younger individuals. MET is an active area of therapeutic and diagnostic investigation in lung cancer, and we found that in our cohort, MET mutations were exclusive to never and light smokers. We confirmed this observation using external cohorts and found that MET mutations were significantly enriched in female never smokers. We also observed that MET was significantly enriched in never smokers over the age of 50 as well. In addition to known splice site mutations, we also identified intronic branch point deletions of MET that are vastly understudied but are known to cause exon 14 skipping, the most well-studied MET mutation. Our custom exome sequencing approach allowed us to capture these mutations. These observations related to MET are novel and exciting and could advance both therapeutic and diagnostic strategies. Oncogenic fusions of ALK, RET, and ROS1 are associated with never smokers. As you can see in this analysis of external cohorts, these fusions are enriched in never smokers. Unexpectedly, however, we found that in our cohort, none of these samples showed any ALK, RET, or ROS1 translocations. We confirmed this using whole genome sequencing as well as targeted AmpliSeq panels. Furthermore, looking at the external cohorts, again, younger never smokers had significantly more fusions compared to older individuals. Additionally, looking at the same external cohorts, we find that fusions are more common in later stage lung cancers. These taken together could be an explanation as to why we did not find any samples with fusions in our cohort, as they are comprised of older women with early stage diagnosis. Next, we went on to perform mutational signature analysis. For this analysis, we compared the mutational signature pattern in our cohort to known signatures that were found in other published lung cancer studies. This figure clusters samples by their normalized signature contributions. 5 distinct clusters are identified. 2 clock signature clusters, a DNA damage response signature cluster, an Agerbeck signature cluster, and a tobacco smoking signature cluster. The tobacco cluster mainly includes heavy smokers, while other clusters are mostly never and light smokers. The clock signature clusters differ. One shows high contributions from both SPS1 and SPS5. The other has contributions from only SPS1, suggesting different mutagenic mechanisms. No somatic or germline mutations were found to drive the DDR signatures. We then looked to see if the absolute number of mutations contributed by clock and Apobeck signatures were different between never and heavy smokers. We found there was no significant difference between never or light and heavy smokers in the number of mutations that could be attributed to either Apobeck or clock signatures. We next investigated whether mutation spectra in oncogenes, specifically EGFR and KRAS, differ between never smokers and smokers. In our cohort and external data, EGFR indel mutations, example exon 19 deletion, exon 20 insertions, are more common in never and light smokers than in heavy smokers. While this suggests more indels in never and light smokers, total number of indels did not significantly differ between different EGFR mutation types. L858R missense mutation in EGFR showed a higher proportion of clock mutations, suggesting clock-driven mechanisms. Indel mutations were significantly enriched in younger patients but showed no sex-based differences. These observations have diagnostic and therapeutic relevance as EGFR indel and missense mutations respond differently to treatment. KRAS mutations are more common in heavy smokers, but KRAS G12 mutations are also present in never and light smokers in our cohort. Most never and light smokers with KRAS G12 mutations showed no tobacco smoke signature, indicating a smoking independent mechanism for these mutations. G12C mutations are enriched in heavy smokers, while G12D and other G12 mutations are more common in never and light smokers. Sex and age do not affect G12 mutations. These observations highlight the need for targeted screening and development of inhibitors for different KRAS mutations, especially in light of the new G12C inhibitors. Next, we wanted to understand the difference in the copy number landscape between these two smoking cohorts. We used Titan to determine the copy number alterations in this cohort. We examined copy number differences between never and light and heavy smokers in our cohort and external cohorts, finding no significant difference in ploidy, fraction genome altered, or whole genome duplication. Sex, age, and pack years also showed no significant impact on these variables. Overall, both smoking groups displayed a very similar pattern of copy number alteration. Recent work by the NCI Sherlock Group identified clusters based on arm level copy number patterns. We next aim to see if similar patterns appeared in our cohort and varied by smoking status. Like the Sherlock study, we found three copy number groups: high arm level deletion group, no arm level alterations, high arm level amplifications. Smoking history did not significantly affect group occupancy. This suggests copy number alterations are likely characteristic of lung cancer itself rather than history of smoking. With that, we come to the end of the results shown in this paper. I would now like to quickly summarize the unique findings from this paper. Mutations in the tumor suppressor MGA may be associated with a history of smoking. MET mutations are enriched in never and light smokers. ApoBec, clock, and DDR signatures predominate the mutational landscape of never smokers. EGFR mutations in never and light smokers are predominantly of the indel type. KRAS G12 mutations are varied in their amino acids in never smokers, and no significant difference is found in copy number alterations based on history of smoking. With that, I would like to thank you all for your attention, and we hope you enjoyed our work.